Thank you very much, Mr. Chairman, and thank you again to Professor Ronco for this kind invitation. So we're going to talk today about a concept that is rather novel, so I ask all of you to try to think a little bit outside the box here with me. These are my disclosures to some funding, but essentially the basic question is that I'm going to pose to you is, how do hosts defend from infection? And we've traditionally understood this, and forgive me for the simplified version of the scheme, but basically the paradigm renders that there's an infection, bacterial overgrowth, and of course there's an immune response that is triggered. And we're going to call that immune response right now resistance, which is going to be the capacity of the host to control that bacterial burden, to eliminate bacteria from the body. And we all know that that tries to decrease this bacterial overgrowth, and by those means try to decrease tissue damage and decrease organ dysfunction. However, we also know that this doesn't come without a cost. And that cost is essentially the production of a lot of cytokines, damage-associated molecular patterns that necessarily also cause damage to the same host that is trying to protect, correct? And this has been called collectively as immunopathology. And we have adjuncts to try to boost those mechanisms of defense. And you probably are aware of many, many trials trying to, again, enhance the capability of drugs that can actually limit the injury that the immune system can potentially cause to the host. However, ecologists have thought of this problem in a different way. It turns out that plants get infected. And they've studied this for a long time. And the way they've looked at this is a little bit different than what we have. They've looked at this problem by assessing the relationship between the capacity of the plant to tolerate a certain bacterial load. And just, if you don't know, because I didn't know this, plants also have a sort of immune system that protects them from these infections. So the capacity of tolerating or containing a bacterial load, just as human beings do, versus measures of health fitness. And health fitness is whatever tells you that the plant is doing okay. In plant biology, it appears that seed production is an important thing. So if a plant is decreasing the seed production because it's infected, that means that the health fitness of the plant is decreasing. All right? Get the concept. So let's make an example here. And here we're going to study a population with a certain genotype. This is a population of plants. This is not only one plant, but a population of plants. This is the health fitness of the plant when it's without infection, very healthy. And this is what happens when the plant starts to experience different bacterial loads. As you can see, ecologists can actually derive more than one dimension of protection from this type of analysis. The first of which is, of course, understanding how capable is that plant to contain the bacterial load, which is exactly what I told you before, the resistance capacity, the capacity of the immune system to fight infection. We know that. This is nothing novel. However, they can also understand what is the repercussion of fighting off that infection. What is the cause in terms of loss of health fitness that fighting that infection represents for the plant. And that is given by essentially the slope of that curve. And that is called tolerance. So tolerance is the capacity of the host to limit injury coming from either bacteria or its own immune system trying to protect the plant from it. Do we get that concept? All right, thank you. So let's compare two groups of hosts. Let's say that these hosts have a genotype A, and we know that they have this type of resistance. And let's compare it to another population of plants that have a genotype B. And I would argue here that potentially they have the same resistance, right? Because they're capable of containing about the same bacterial load. But what would you say about the tolerance capacity? Is it different? People are shaking their heads, right? So yes, for B, there's way little less sacrifice in terms of health fitness than for A. And that means that B has a higher tolerance than A. Which plant is going to do better? B, right? And this is completely independent of the capacity of the immune system of the plant to defend itself. You see the power of this concept for potentially translating this into our patients. So the ecology paradigm doesn't pose only one mechanism for defense, but rather two of them. One is the resistance capacity, which is in charge, the immune system is in charge to decrease bacteria, and of course, with the cost of causing immunopathology. But on the other hand, there's tolerance, which is intrinsic mechanisms of the host that bolster the capacity of that host to weather this damage that is caused by immunopathology. If we could harness that, that would be yet another strategy to try to protect organs while the immune system is doing whatever it needs to do to clear bacteria. Okay, so who cares about plants? Does this happen in mammals? And Raberg was the first one, and look at the date. This is a newborn concept for our field. Raberg was the first to actually demonstrate this in a model of malaria. What he did was infect 5 different genotypes of animals with malaria, and then he looked at these same relationships. And he looked at the peak parasite density versus the level of anemia as a measure of health fitness. That makes sense because malaria causes anemia, and the deeper the anemia is, the worse the severity of malaria is, and the higher the mortality is. So what he found was that exactly what the ecologists have found with plants, that each of these genotypes of animals had a different tolerance capacity based on the fact that they had different slopes in this relationship. This was the first time that this was demonstrated in mammals. So what are the mechanisms that could potentially bolster tolerance capacity? And this is where the field is ripe for investigation because we have no clue. In plants, they might have some idea of what the mechanisms are, but in our field, very little is known, and just a few groups have started to do some work in this field. But I'll give you an example in sepsis, actually. It turns out that sepsis also causes hemolysis and liberates free hemoglobin as well as heme that through the production of reactive oxygen species and production of DAMPs causes cell tissue damage. Well, Larsen did a very, very interesting study where they found that the expression of heme oxygenase, which is an enzyme necessary to deal with the digestion of this heme, was protective in this setting because animals that lacked HO1 or heme oxygenase 1 died more when infected during experimental sepsis. And furthermore, they looked at what was the impact of expressing or not expressing heme oxygenase in the level of bacteria in the peritoneum after cecal ligation and puncture, which is a measure of how good your immune system is being able to clear bacteria. And what they found was that there was no difference. Therefore, this was an entirely tolerance mechanism that was protecting the hosts from death. They further looked at another molecule that actually is produced in the body, which is hemopexin, that binds to heme. And what they did was give recombinant hemopexin to these animals and hypothesize that that would protect the animals as well. And lo and behold, yes, there was protection when they gave these animals hemopexin. Again, not having anything to do with the capacity of the animal to get rid of bacteria. So this is a potential tolerance mechanism that can be harnessed and can be further evaluated. Is this pertinent to septic patients? This same group actually looked at septic patients and measured levels of hemopexin. And interestingly, they showed an inverse relationship between the level of hemopexin and mortality in such a way that non-survivors had lower hemopexin levels than those patients that went on to survive. Now, of course, this is a long way from us saying, well, hemopexin is the fix for sepsis. That's not what I'm trying to say. I'm just trying to say that this is a mechanism that is completely independent of whether or not you're giving the right antibiotic, and it's showing protection in animals and potentially in human beings. But no good deed goes unpunished. Of course, the digestion of heme by heme oxygenase 1 can also produce labile iron that can go ahead and produce reactive oxygen species. And it's been shown, this group by Wise et al., that that production of reactive oxygen species inhibits glucose 6-phosphatase, which is the key enzyme in the liver to produce gluconeogenesis. So when they looked at this, they were seeing that the animals were actually dying of hypoglycemia because their livers were not able to sustain glucose production through gluconeogenesis. And they argued that perhaps the expression of ferritin, which is an acute reactant produced in the liver, could be protective. And what they showed in animals that do not express ferritin by knockout mechanisms versus those that do, they showed a substantial difference in mortality. Again, not related to the levels of bacteria cleared from either the peritoneal cavity, blood, liver, or spleen, again, arguing that this might be a tolerance mechanism. So this was the little cartoon that they presented, and their thought process was that ferritin decreases the development of reactive oxygen species, and that enables glucose 6-phosphatase to actually act, do gluconeogenesis in the liver and sustain glucose levels. However, our patients basically never die of hypoglycemia. And we were interested in another piece of data that they presented, which was by metabolomic analysis, they try to understand whether these animals were actually utilizing oxidative phosphorylation as their main source of energy. And just to remind you, glucose can either be metabolized through the glycolytic pathway, which I've shortened dramatically here, or by shuttling pyruvate into the mitochondria and going to a Krebs cycle, and then to the mitochondria, to the mitochondrial electron transport chain to utilize as through oxidative phosphorylation. And I want to point your attention towards these four pieces and to the white bars here, which are the animals that were wild types. They were completely normal animals. And what you can see by 48 hours is that these animals actually all are showing incremental levels of these intermediaries of the Krebs cycle, suggesting that they were all using more oxidative phosphorylation than glycolysis, whereas the animals that did not have ferritin had the opposite direction, suggesting that they were using mostly glycolysis. You might be asking, why am I telling you this? And I'm going to try to show you why this is important. It turns out that sepsis, if you take monocytes and you stimulate them with lipopolysaccharide, which is a model of in vitro sepsis, what these monocytes do is they decrease the oxidation of glucose and they increase the utilization of aerobic glycolysis. Exactly what I was showing you before in the previous slide. And later on, this change in metabolism is followed by a return to oxidative phosphorylation, as you can see here, by an increase in the uptake of fatty acids and an increase in fatty acid oxidation. These are the drivers of these processes that have been shown in monocytes. HIF1 alpha is in charge to actually switching metabolism to glycolysis by expression of these enzymes right here, pyruvate kinase M2, lactate dehydrogenase and pyruvate dehydrogenase kinase, and SIRT1, the NAD-dependent deacetylases, have been charged with the turnaround towards oxidative phosphorylation. Importantly, changes in metabolism change how these cells act and how these cells behave, because if they switch to glycolysis, they become pro-inflammatory, whereas when they switch back to oxidative phosphorylation, they act as if they were anti-inflammatory, as if that was the knob that the cell needed to switch in order to turn off inflammation, for example. And furthermore, it's been shown that if you block this switch to glycolysis in the setting of sepsis, using, for example, chiconin, which is an inhibitor of PKM2, you improve mortality. Well, these are monocytes. What about the kidney? So we've looked at this, and our hypothesis was that perhaps the kidney behaved in a similar way. And we looked at what would happen to renal tubular cells in culture exposed to lipopolysaccharide in terms of the utilization of glycolysis and oxidative phosphorylation, thinking that they had the same biphasic behavior. And this is a little bit of a complicated slide, but chart, but I'll walk you through it. This extracellular acidification rate is a surrogate for utilization of glycolysis, and oxygen consumption rate is a surrogate for utilization of oxidative phosphorylation. And I want you to focus on these two. And what happened was that from 3 hours to 24 hours, these cells started decreasing the utilization of glycolysis and increasing the utilization of oxidative phosphorylation, again suggesting that there was this biphasic way. And the drivers behaved exactly the same. HIF1 alpha and PDHK rose initially and fell towards 24 hours, and SIRT1, which is the driver of oxidative phosphorylation, rose at around 24 hours. So it seems like this happens in cells. What happens in animals? We did metabolomic analysis and showed that 8 hours after cecal ligation and puncture in kidneys of animals exposed to this, there's a switch towards glycolysis and a decrease in oxidative phosphorylation. So it seems like this mechanism is apparently working in the setting of sepsis and in the kidney. So in my previous talk yesterday, I told you that we have shown that AMP-activated protein kinase, which is a master regulator of energy, is activated during sepsis. And what I want to show you now is that if you enhance the activation of this energy regulator, you protect the kidney from AKI in the context of cecal ligation and puncture-induced sepsis. And furthermore, you increase the probability of survival of these animals. And I'm showing you here data using ACAR as an activator and using metformin as an activator of AMPK. And furthermore, when we looked at the bacterial levels in the peritoneum of these animals, we found that there was no difference between the animals that did not receive and did receive the activator of AMPK. Again, suggesting that this mechanism through AMPK activation could be a tolerance mechanism. And this is the importance of metabolic reprogramming as a defense strategy of the kidney in the setting of inflammation. I just want to point out another mechanism which is dependent on the inactivation of the mammalian target of rapamycin, which is the natural inhibitor of autophagy. Autophagy is a process by which damaged organelles and mitochondria are targeted and engulfed in autophagosomes to be degraded and then reproduced in newer functional mitochondria. So Figueiredo et al. Actually look at anthracyclines, which are anti-cancer medication, to actually treat sepsis, which is a little bit weird. But they ended up showing that these anthracyclines work through inhibition of the mammalian target of rapamycin and only protect the kidney when autophagy is functional. Because in animals that had no autophagy, this medication did not protect against survival. And again, showed that there was no difference between the bacterial load in the peritoneum, suggesting again that mitophagy or autophagy activation during sepsis might be a tolerance mechanism. One example on why to be cautious. Wang et al. Just recently published a paper that tried to assess whether anorexia was a tolerance mechanism of protection, right? When we all get sick, we don't want to eat. We just want to lay down, right? Well, and they compared animals exposed to bacterial infections versus viral infections. This is what happened. In bacterial infections, animals that were exposed to anorexia lived more than those that were fed. Look at what happened in animals that were infected with viruses. Exactly the opposite. So we got to be very cautious because this is going to be very particular to the host, to the type of infection, and probably to the situation that the patient, the mouse, the plant is living in, and the environment that is touching it. So with that, I think maybe a new paradigm is in order. And I put new in quotes because ecologists would laugh at us just to call this new. In essence, tolerance is a new mechanism that could potentially leverage cell responses to inflammation to protect from tissue damage and potentially protect from organ dysfunction. And just to conclude, resistance and tolerance are two independent but complementary defense strategies active in mammals and humans. There are many tolerance mechanisms. Most of them are unknown. The field is ripe for investigation. Metabolic reprogramming is key in the response to sepsis, impacting organ function, repair, and outcome, and possibly orchestrating some of these tolerance mechanisms. Tolerance mechanisms are specific to the host, disease process, and infectious agent. This is not something that we need. We want to just hop on to do the next clinical trial with one magic drug and it doesn't work. We got to understand these mechanisms better before doing that. And then further efforts are definitely needed to understand these mechanisms better, to leverage novel avenues for phenotyping, diagnosing, and treating our patients. And with that, I thank you for your attention.